You may have noticed that we've talked about how life changes, but we haven't yet talked about the origins of life. And that's what I'm going to do now. But the reason why with this threshold we didn't begin with the origins is that it's vital to understand how life changes. In fact, it's vital to understand the mechanism of natural selection before we get a sense of how life originated. Because in modern understanding of the origins of life, natural selection itself plays a crucial role. Now, I'm going to come at the subject from a slightly different angle. Let's talk about Fred Hoyle, who was one of the greatest of modern British astronomers. His dates are 1915 to 2001. Fred Hoyle's a very important figure in several modern scientific disciplines. He pioneered our modern understanding of how chemicals are formed in stars and supernovae. So he could play a starring role in the lecture on the formation of elements. But Fred Hoyle was also famous for being, what shall we say, a contrarian. That is to say, he was someone who seemed to enjoy taking positions that most scientists opposed. For example, he never accepted the Big Bang Theory, despite being one of the best-known, most influential physicists of his generation. And he defended modified versions of his steady-state theory. That's the theory that the expansion of the universe was caused by the continual creation of matter in space. He defended his steady-state theory throughout his life. He also refused to accept that life had evolved on Earth, that life had originated on Earth. And he claimed it must have evolved elsewhere and arrived in the form of a rain of living material from space. This was the theory known as panspermia. Fred Hoyle, in connection with this, wrote a wonderful science fiction novel, which I remember seeing in televised form as a teenager in the early 1960s. It was called A for Andromeda. In A for Andromeda, British astronomers, of course British, receive a signal from space, and they realize that it's got a pattern to it, and eventually they decide it's a sort of code. And they look at the code, and after time they crack it, and they realize it looks like instructions for building a computer. So they build the computer, and then they press the start button, only to find out that the goal of the computer is to construct a new race of alien beings. This is a very elegant way of moving life from one part of the universe to another. I should confess at this point that my very happy memories of this series probably coloured by the fact that the first alien created by the machine was played by a young Julie Christie. Now, Hoyle described his primary objection to the idea that life had evolved on Earth as follows, in a very famous metaphor. This is what he said. A junkyard contains all the bits and pieces of a Boeing 747, dismembered and in disarray. A whirlwind happens to blow through the yard. What is the chance that after its passage, a fully assembled 747, ready to fly, will be found standing there? Now, Hoyle is supposed to have made the comparison with a 747 because yeast cells and Boeing 747s both contain about 6 million parts. In some sense, we could say they represent similar levels of complexity. Fred Hoyle's objection to the idea that life could have evolved on Earth through blind processes such as some form of natural selection express a widely held view that life is simply too complex to have been the product of blind random processes such as those that drive natural selection. His own answer, we've seen, was that life must have evolved somewhere else. And quite frankly, I hope you'll agree that this is really, in its own way, very unsatisfactory. And it's a bit like announcing the existence of a creator God. It simply postpones the question by forcing us to try and figure out how it could have formed somewhere else in the universe. So this is the question. Can we do better in explaining the origins of life? Most biologists today think we can do a lot better. And in this lecture, I'm going to try to describe their efforts to do so, to explain the origins of life on Earth in modern scientific terms. Now, the last lecture described why most biologists accept that natural selection explains the variety and adaptability of life today. But the question is, can natural selection also help us explain the initial appearance of life? Can it overcome the objections of critics like Hoyle? I'm going to try and show that it can. So how were the first life forms created? That's a question that Darwin himself seems to have had no answers to. A little speculation, but no serious answers. I've deliberately held off with this question, as we've seen, because we do need a good grasp of the mechanism of natural selection before we can understand the modern answer. It has to be said, though, that the modern answer is not yet quite as good as many biologists would like it to be. We have a very good answer. We have lots of pieces of the puzzle. But I suspect many biologists would agree we don't have quite as complete a scientific theory of the origins of life as they would like. But nevertheless, we can offer a very good attempt at this. And we can offer a very good answer to Hoyle. Okay. As with earlier levels of complexity, it'll help if I put modern explanations in context by comparing them with the answers adopted in traditional societies or pre-scientific traditions. How have traditional societies explained the appearance of life? We can divide traditional explanations into two broad categories, the divine and the naturalistic. Divine models by now we're familiar with. They're deistic. They assume that life was created by a divine being. And we've seen the problems with such theories over and over again, and we've seen why they're not really admissible within modern science. Modern science does not assert that these theories are wrong, merely that they are untestable and therefore do not count as science. What are naturalistic models? Well, naturalistic models have a long history, and they are in fact attempts to argue that life, the appearance of life, can be explained without necessarily assuming that they were created directly by a god of some kind, that life can be generated spontaneously from existing materials and forces. And this implies, of course, that life is not so utterly different from the rest of the world. There's a sort of continuum between life and non-life, just as Newton implied that there's actually a continuum between the heavenly realm and the earthly realm. This is how Aristotle put it. Aristotle was a committed believer in the possibility of the spontaneous generation of life. He wrote, "The transition by which nature passes from lifeless things to animal life is so insensible that one can determine no exact line of demarcation, nor say for certain on which side any intermediate form should be. Thus, nature passes from lifeless things to animals in an unbroken sequence." For traditional scientists like Aristotle, the evidence for spontaneous generation was things like the appearance of maggots in rotting meat. If you feel like it, you can try this as an experiment. It's very, very cheap and it's very easy to conduct. And this is the recipe as told to me by my colleague David Briscoe, who lectured on life in the early versions of my big history course, and his lectures were wonderful. This is his recipe. He says, take a soup. There must be something meaty in it. Preferably don't take a very good soup, and David used to conventionally recommend one of his own. You stick it outside, you leave it in a warm, exposed place for a few days. Voila! Maggots and all sorts of other creepy crawlies. So there's the evidence for spontaneous generation. It's a nice idea. It's sort of commonsensical, a bit like saying that the Earth is obviously not moving because we don't feel the wind rushing past. But unfortunately, of course, it's not quite that simple. From the time of the scientific revolution, scientists began to try to test such ideas much more carefully, as scientists became more and more committed to ensuring that their theories were based on hard evidence. And the attempt to test the idea of spontaneous generation led to some interesting toing and froing on the issue. This is a long story, and I'm just going to pick out some highlights. In 1765, Lazzaro Spallanzani, an Italian biologist who lived from 1729 to 1799, tried to test Aristotle's idea, and he did it by showing that if you took a meat broth and you boiled it to kill off all the bugs that were already in it, and then you placed it in an airtight container so that no bugs could get into it from outside, nothing appeared. No maggots, no creepy crawlies. Now, this is, to us, very familiar indeed. We all know about sterilization. We expect jars in supermarkets to be sterilized, so this is all very, very familiar. But what did he actually show? What did Spallanzani actually show? He showed that the maggots must have come from something outside the broth itself, probably from eggs planted by their parents. So this seemed to suggest that maybe spontaneous generation was not possible. Maybe every living thing has a parent. All living things come from other living things. Now, Spallanzani didn't have it all his own way. He had opponents who suggested an alternative, and this was that what he'd actually done by insulating his container was to exclude the life force that was necessary for the generation of life. What they had in mind seems to have been something like this. It was the idea that there was some special force in the air that got into things and made them alive. Now, we've already seen a similar theory, which was refuted as early as 1828, and that was the idea that there were some chemicals, organic chemicals, that contained a sort of principle of life, and that they were necessary for the creation of life. And we saw that in 1828, Friedrich Wöhler had disproved that idea by synthesizing simple organic chemicals in the lab. But here's the second version of that theory. It is that maybe there's a life force floating around us in the air, and it needs to get in to a broth to create life. Now, how can you test such an idea? It's an interesting idea. How could you prove it's right or wrong? Testing this idea quite decisively was one of the great achievements of the French chemist Louis Pasteur. And his experiment is one of the most elegant of all scientific experiments. Very, very simple, but he provided a wonderful test for this idea. This is what he did. In 1862, Louis Pasteur's dates, by the way, are 1822 to 1895, and he's an immensely important figure in modern chemistry. What he did was he created an apparatus which began with a glass flask. At the top, there was a long glass tube. Now, it came out the top, then he bent it back down to the ground in a sort of U-bend, and then bent it up again. And he left the opening open. He didn't plug it, he didn't close it at all. So it's open to the air. He put a sort of broth inside the flask, and then he heated it to sterilize it. He boiled it to sterilize it. And then he just left it. That's the experiment. Now, this is the idea. If life actually came from pre-existing life, from pollen or from spores or from tiny organisms floating in the air, then his argument was that such organisms could drop into the spout, but they'd get caught in the U-bend. They wouldn't be able to get into the broth, so no life would appear. If, on the contrary, life came from a life force, now surely the life force would be able to travel down the tube, up the U-bend, and get into the broth. So he boiled his flasks and waited. And what happened? I'm told, though I've not been there myself, that you can still see his flasks in the Pasteur Institute in Paris, and that they're still as sterile as the day when he first boiled them. That seemed to be the end of the story. That was more or less the end of the theory of a free-floating life force of some kind. And it also seemed to have put paid to the whole notion of spontaneous generation. That, of course, created a new problem. Pasteur seemed to have proved that life could only come from previous life, from eggs or spores. Spontaneous generation was impossible. If so, how were the first living organisms created? In the early 20th century, biologists faced a real dilemma because he seemed to have proved that spontaneous generation was not possible. How can you explain the origins of life? Well, now I want to move towards modern explanations of this. Biologists began to get some purchase on this knotty problem in the first half of the 20th century. And I'm going to describe their explanations in 3 stages. Now, let me say immediately, there are a lot of technicalities here, so this is an extremely simple description of some very, very complex and elaborate arguments. But nevertheless, I think we can capture the essence of the arguments. The first stage, let me summarize the stages here. The first stage is the creation of the simple chemicals, each with only a few atoms, that were necessary for the creation of life. Chemicals such as amino acids that form proteins and nucleic acids that are the basis of the genetic material of all living organisms. That's the first stage. The second stage is the creation from these simple components of huge, complex molecules, mostly taking the form of long, long chains that form the molecules that we find in living cells. That's the second stage. The third stage, and at present the thorniest, is to explain the creation of the modern mechanism of reproduction, which is based on the extraordinarily complex molecule DNA, or deoxyribonucleic acid. Let's take each of these steps in turn. So first, the creation of the simple chemicals of life. These are chemicals that consist of just a few atoms. They're present in all living organisms, such as the amino acids that make proteins, the nucleic acids that are the basis of DNA, our genetic material, carbohydrates that make sugars and starches and act as sort of stores of energy, and the lipids that make fats and hormones and membranes. How can you make these simple chemicals? So we need to explain the creation of the chemicals vital for life. These are the structural basis for most living organisms. Today, atmospheric oxygen destroys such molecules. We've seen that oxygen is fantastically reactive. And that's why, as Pasteur claimed, life can no longer be generated spontaneously. With an oxygen-rich atmosphere, it seems as if life cannot even get off the ground because you cannot make even the simplest chemicals of life. That was the real problem scientists faced in the early 20th century. However, in the 1920s, Alexander Oparin in Russia and J.B.S. Haldane in Britain pointed out a solution. And this was that maybe these chemicals could have been formed spontaneously in an atmosphere that didn't have free oxygen in it. So where will you find such an atmosphere? Now, I hope by now you already know the answer to this. The answer was in the atmosphere of the early Earth. We saw that free oxygen didn't start building up in the Earth's atmosphere until about 2.5 billion years ago. So this means that though we wouldn't have liked the early Earth, it turns out that in fact it might have been a more benign environment than today's Earth for the very simplest and earliest stages of life. Now, once again, let's ask the question, how would you test such an idea? And again, we can describe a beautiful, simple experiment that tested it very well indeed. And it's associated with the work of Stanley Miller, a graduate student who worked under the supervision of a very well-known biologist, Harold Urey. In 1952, Stanley Miller organized a very famous experiment to test the idea that in an oxygen-free environment it might be easy to generate these simple molecules. Let me describe the apparatus for you. So once again, we've got a glass flask. Coming out of it is a tube which loops around in a square and comes back into the flask. He fills the glass tube with gases that might have been present in the early atmosphere, and there's no free oxygen amongst them. He used methane, ammonia, and hydrogen, but later versions have used many other mixtures. He then added water and supplied energy in the form of heat from below and electric charges. Now, why water? Why was water so crucial? Well, why do biologists assume that any planet with life must have water in liquid form? Here's the naive non-chemist's answer. And we've briefly seen this before. Water provides an ideal environment for complex chemical reactions. In gases, Molecules are charging around at huge distances from each other. They encounter each other actually quite rarely, and when they do, it's very violent, and they often bounce apart. In solids, atoms are locked in and they barely move. So there's not much possibility for rearranging themselves there. Not much possibility for interesting chemical things to happen. In liquids, atoms and molecules can move close to each other, but they also have a lot of freedom to move around. A bit like dancers on a dance floor. And water itself provides a chemically benign atmosphere for such movements, as it's not itself terribly reactive. So this is why water was so crucial, and this is why water is probably always crucial in the creation of life. Okay, now what happened inside Miller's apparatus? He leaves it humming there for a few days. And then after a few days, a dark red sludge appears. And when you take the sludge out and you analyze it, you find it contains amino acids, nucleotides, and phospholipids. Lipids, the chemicals you need to form membranes. This experiment has been repeated many, many times. And it's now very, very clear, it's been repeated using different gases, that creating these simple chemicals is really not a huge problem in an environment without free oxygen. So that's the first stage. And it seems that that was fairly simple. We can explain the creation of these very simple molecules consisting of just a few atoms or a few tens of atoms from which living things are made. Now, there's a second step. How do you take those and build from them huge, elaborate molecules of the kind that you find in living cells? The Urey-Miller experiment produced just these very simple molecules. How do you take the next stage? Even the simplest viruses contain billions of atoms in very complex configurations. And many of these molecules take the form of huge chains of atoms arranged in very specific ways. And along those chains, you'll find tiny electric charges, so that the chains tend to sort of wrap up on themselves, forming complex balls with very specific shapes. And very often the specific shapes they form determine the precise chemical or physical role they play within them. So the exact sequence of chemicals along them is vital. Now, how can you create such huge and complex molecules? Well, not by chance, and that was the point that Fred Hoyle was trying to make in the example I've given already about whirlwinds and Boeings. Hoyle made the comparisons as we've seen because yeast cells and Boeings both exist at about the same level of complexity. So how can we get past the riddle that Hoyle posed? Fortunately, there is an answer, and this takes us back to natural selection. Though random changes, imagine all these chemicals sloshing around within a liquid environment. Random changes are unlikely to create perfect long molecules. But let's imagine something else. We add a new rule, which is that if each successful step towards life can be locked into place, then the odds improve drastically. Now, this is precisely how natural selection works. By locking into place random variations that create something viable and stable. So here's the idea, and it's a central idea in modern biology. It is that some complex chemicals, or chemicals that are becoming more complex, can actually evolve through a chemical version of natural selection. This is the idea of chemical evolution. If you get chemicals that have a certain stability, well, they're going to survive. And they're going to be on the dance floor, and they're going to mix with other chemicals so that slowly you can build up complexity. In the 1950s and 1960s, Sidney Fox showed how chemical evolution might work. He showed some of the specific pathways that might be taken. For example, under certain conditions, long chains form spontaneously, similar to those in living organisms. Some of these long molecules naturally curl up to form spheres that are beginning to look a bit like simple cells. And some of them have semi-permeable membranes through which they can take in chemicals from the outside and they can get rid of stuff they don't need, that they reject. So we already have things that are beginning to look a bit like very simple cells. They have a metabolism, they can take in energy and food from outside, they have the ability to reproduce because some of these, when they get big enough, simply split. They simply break apart. And they can also adapt over time. This gives them all of the emergent properties of life in a sort of simple and rough and ready form. So here we have a sort of chemical proto-life. Now, while we're on the subject, where did the earliest organisms appear? Where did this sort of chemistry probably take place? Where was the dance floor for all of this? Darwin assumed that life first appeared in what he called some warm pond. He knew it had to be liquid. He knew there'd have to be some warmth, some energy. He understood all of that perfectly well. But now it's generally thought that the surface of the Earth would have been a dangerous and hostile place for early life. So a more likely environment may be these deep-sea volcanic vents that we described in describing modern plate tectonics. Ideal conditions for chemical evolution probably existed near these mid-oceanic vents. There was lots of energy as magma poured up from the interior of the Earth. There was a rich mixture of organic chemicals, again, coming up from the inside of the Earth. And there was protection from ultraviolet radiation because you're deep inside the oceans, you're way below the surface. And it's been discovered that there are very rich colonies of chemical-eating bacteria around mid-oceanic vents. So at the moment, this looks like our chemical dance floor. That's stage two. We can explain the creation of large, elaborate chemicals, and some of them are beginning to look a bit like simple cells. Now, stage three. It's actually the trickiest part of all. How do we explain the creation of the most complex molecules of all, those involved in reproduction? DNA, deoxyribonucleic acid. DNA is the software that controls reproduction, and explaining how DNA evolved is really the trickiest part of the puzzle. Without DNA, reproduction is bound to be inaccurate, and that means that chemical evolution would have been slow and unreliable. It wouldn't have been very good at preserving valuable variations, adaptive changes or mutations. What's DNA like? Let me try very quickly to try and describe it. You'll all be familiar with pictures. DNA consists of two linked chains of nucleotides, millions of these nucleotides. These are little groups of clusters of atoms. And each DNA molecule consists of two of these chains. They're linked with bonds like rungs on a ladder. Now, it's the bonds that are the really interesting part of DNA. Each bond consists of two bases. These are small clusters of atoms. That's all we need to know. The bases come in four main types, and they can only fit together in certain ways. The four types are A, T, C, and G, which stand for adenine, thymine, cytosine, and guanine. And this is the rule. A links only with T, and C only with G. So along each of these rungs, you have, sticking out from one chain, maybe an A, and it's linking with a T, which is sticking out from the other chain. Now, imagine we separate the two. We run along one of the chains. What we find is a long list of bases, A, T, C, G. That is the code. That's the genetic code. The exact sequence of those bases contains the code for creating a living organism. How does DNA reproduce? It splits down the middle. All the rungs split. Each chain goes off in search of its pairs. So if you have a rung with A, it looks for T, and so on, all the way down. And eventually it reassembles its complement. So you now have two identical chains, each forming a helix. This is the key to accurate reproduction. The trouble is that DNA cannot exist on its own. It's like software. Yet cells can't survive without DNA. So which evolved first? The software of DNA or the hardware of the cell? It's a puzzle. And at present, all we can say is that the best bet is that RNA, ribonucleic acid, which is closely related to DNA, may have acted at one time as both hardware and software. RNA comes in single chains, so it can do the work of software, but it can also do the work of proteins within cells. It can do both. So there are three stages: forming the basic chemicals, turning them into large complex molecules, and creating DNA, the basic genetic structure. Now, I hope I've conveyed a sense that we know a lot of this story. I shouldn't say we, the biologists know a lot of the story. They know a lot of the details, but it's still not quite perfect. Nevertheless, there's enough here for biologists to be very confident that they can essentially explain how living things were assembled from non-living ingredients about 3.5 billion years ago, or slightly more. And that's something we'll look at in the next lecture. Thank you. You may have noticed that we've talked about how life changes, but we haven't yet talked about the origins of life. And that's what I'm going to do now. But the reason why with this threshold we didn't begin with the origins is that it's vital to understand how life changes. In fact, it's vital to understand the mechanism of natural selection before we get a sense of how life originated. Because in modern understanding of the origins of life, natural selection, for example, he never accepted the Big Bang Theory, despite being one of the best-known, most influential physicists of his generation. And he defended modified versions of his steady-state theory. That's the theory that the expansion of the universe was caused by the continual creation of matter in space. He defended his steady-state theory throughout his life. He also refused to accept that life had evolved on Earth, that life had originated on Earth. And he claimed it must have evolved elsewhere and arrived in the form of a rain of living material from space. This was the theory known as panspermia. Fred Hoyle, in connection with this, wrote a wonderful science fiction novel, which I remember seeing in televised form as a teenager in the early 1960s. It was called A for Andromeda. In A for Andromeda, British astronomers, of course British, receive a signal from space, and they realize that it's got a pattern to it, and eventually they decide it's a sort of code. And they look at the code, and after time they crack it, and they realize it looks like instructions for building a computer. So they build the computer, and then they press the start button, only to find out that the goal of the computer is to construct a new race of alien beings. This is a very elegant way of moving life. Self plays a crucial role. Now, I'm going to come at the subject from a slightly different angle. Let's talk about Fred Hoyle, who was one of the greatest of modern British astronomers. His dates are 1915 to 2001. Fred Hoyle's a very important figure in several modern scientific disciplines. He pioneered our modern understanding of how chemicals are formed in stars and supernovae. So he could play a starring role in the lecture on the formation of elements. But Fred Hoyle was also famous for being, what shall we say, a contrarian. That is to say, he was someone who seemed to enjoy taking positions that most scientists opposed, from one part of the universe to another. I should confess at this point that my very happy memories of this series probably coloured by the fact that the first alien created by the machine was played by a young Julie Christie. Now, Hoyle described his primary objection to the idea that life had evolved on Earth as follows, in a very famous metaphor. This is what he said. A junkyard contains all the bits and pieces of a Boeing 747, dismembered and in disarray. A whirlwind happens to blow through the yard. What is the chance that after its passage, a fully assembled 747, ready to fly, will be found standing there.